All right, class. We are talking about viruses this unit. First, I just wanted to point out the size of the viruses. Compared to a prokaryotic or a bacteria cell and a eukaryotic cell, viruses are extremely small and extremely simple. A virus is only 20 to 300 nanometers. That's really tiny. That's so small you can't see it on a light microscope. Whereas a bacteria is somewhere around 5,000 nanometers. And if you do the math, that's significantly bigger than the virus. And then you've got the eukaryotic cell. Eukaryotic cell is way bigger. Now, once again, this is a range and it's average, but the virus is teeny, teeny, tiny compared to the eukaryotic cell. In fact, the ribosome that you would find in a bacteria or eukaryotic cell is about 25 nanometers. And there are actually viruses that are smaller than a ribosome. Now, as far as virus structure is concerned, this is what our project, our main push, is focused on. All viruses have genetic material, so they all have either DNA or RNA. And that DNA or RNA could be double-stranded or it could be single-stranded. All viruses have a shell around their genetic material called a capsid, and that capsid is made of proteins called capsomeres. Okay, and it's almost like beading, like the capsomeres kind of form like a protective shield around that genetic material. Some viruses will have an additional layer similar to a cell's membrane, and they get that from the actual host cells that they infect. And the outermost layer of a virus will have proteins for IDing target cells or host cells, and also for latching on or attaching to them. And then some viruses will have a few enzymes inside of them, and that like the RNA viruses, and that helps them turn their RNA into DNA once they get into the cell.
Okay, this slide right here is going to show the different sizes of living things compared to a virus. So if you look in the top left corner right here, that is a red blood cell, and that red blood cell is way bigger than this tiny little prokaryote. And then this tiny little smudge you see right here, those are viruses. So viruses are teeny, teeny, tiny. Now if you take this bacteria cell and you zoom in real close, this is E. coli. You can see it takes up this whole screen. Now you can see the little viruses a little bit more clear. These are the smudges. Now we zoom in more on the viruses, and you can tell here's the bacteriophage. It is actually what infects E. coli. Very small. The smallpox virus looks like this. This is a virus that attacks tobacco plants. Here's another bacteriophage that would infect something like E. coli. Here's polio. Now this little guy right here is the reason why many people early in the 1900s lost the ability to walk. Very, very bad virus, but just the emphasis here is on size. Look at the size of this virus compared to, say, like a ribosome right here. That all it does is assemble proteins. It's a small, tiny part of a bacteria cell, and the virus is barely bigger than that. Okay, we're going to look at the next slide here. Here are some structures. The key thing I want y'all to focus on is that there are two major structures in all of the viruses. You've got the DNA and RNA. That's the genetic material. Like right here, this adenovirus has DNA, whereas this tobacco mosaic virus has RNA. So it's got some sort of genetic material. All four of these different shapes do. They also have a capsid. And the capsid is basically a coat of proteins, kind of like a shield or armor that protects the genetic material on the inside. And they're made up of these little proteins called capsomeres. So a lot of these capsomeres link together, kind of like Legos, that form a shield to protect. Now, once the virus gets into, especially these two right here, once these viruses get into a cell, a host cell, the capsid will dissolve and release the genetic material to do its job. Over here, this one is unique, and it is a bacteriophage, and this is one of the bacteriophages that attacks E. coli. And its capsid will actually stay on the outside of the cell. And I don't know if you can notice here, but there are some spiky things here, and these legs will grab onto the E. coli bacteria, and then it'll press down, and it'll actually inject its genetic material into the cell, leaving the shell on the outside. but the rest of these get absorbed completely into their host cells. All right, and here is just a comparison between the structure of HIV, which is the human immunodeficiency virus, and a typical cell. Now the sizes are not in proportion. Obviously the eukaryotic cell is way bigger, but just look at the complexity of the eukaryotic cell, the nucleus, the DNA, all these little organelles that do work for the cell, and these tiny little dots here, those are ribosomes. Okay, and this virus is not much bigger than one of these little ribosomes, so it's teeny tiny. But as you can tell right here, this has actually a special coating on the outside that is like a cell membrane, so it kind of disguises or camouflages the virus. So our body doesn't always detect the HIV virus, and that's why it's so lethal, because it kind of wears this cloak around it that protects it and disguises it. But inside you've got the capsid, that protein coat that protects the genetic material, and you've got the actual genetic material, and then you've got this enzyme that helps to manufacture the genetic material, the RNA, into DNA once it's infiltrated the host, injected into the host. So that is the structure of viruses: tiny, tiny, small, complex, non-living viruses.